thank you so much to all the assistants for being with us today. I'm Dr. Laura Cordero. I work as medical advisor at MetaCell Technology, and I will be hosting today's webinar. So today's webinar will be focused on autologous exosomes and the role in aesthetic medicine, and we will cover whether are they a game changer or just a trend. And to do so, we will have two esteemed speakers with us, Dr. Sophie Schotter and Dr. Hernan Pinto. And
Hi.
yes, importantly, yes, Hernan?
No, hello to everybody.
Okay. Yes. Okay. So I want to just set very clear two main important points. The first one is that this webinar is intended for licensed healthcare professionals. So please, if you're not a qualified HCP, we kindly ask you to leave the session. The second one is that the webinar will be recorded, and we will send the recording to all the registrants. So no worries, we will send you the whole video. And I think with that, I just had some slides just to set a little bit of the context, and then we will move with the main presentation. So we know that regenerative aesthetics are becoming more popular in aesthetic medicine, and their main aim is to explore the use of biologically active substances in order to support the skin, the hair, and tissues' natural repair and renewal processes. So why are they gaining so much popularity? They are more natural. They try to address the root cause of aging, not just the symptoms, and they want to complement or enhance traditional treatments. And among the most important regenerative aesthetic treatments, we have PRP, fat grafting, but the rising star right now in aesthetic medicine, and specifically in regenerative aesthetics, are exosomes. And in order to talk about this hot topic in aesthetic medicine, we have our two main speakers. The first one is Dr. Sophie Schotter, who is an award-winning aesthetic doctor coming from the UK. She's the founder and medical director of Illuminate Skin Clinics in Kent and London. She's also a media contributor. She has her own podcast, Age Well with Dr. Sophie Schotter. She's won many awards in aesthetic medicine, and she's also an international trainer and speaker. So thank you so much for being with us today. And our second speaker is Dr. Hernan Pinto, who is the Chief Science Officer at MetaCell Technology. He's also the director of the I2E3 Biomedical Research Institute. He's professor at different master courses in aesthetic medicine at different universities in Spain. He's also very invested in publications, research. He's also an international trainer and speaker. So thank you so much, Hernan, for being with us today. And the way the whole session is going to run is that it will last around 1 hour. The presentations of the speakers will last around 45 minutes, and then we will leave a 15-minute slot at the end of the session to have a live discussion and to cover the main questions. So to all the assistants, please, if you have any questions, just use the chat box on the right of the screen and just type there your questions. I'm going to be collecting all of them throughout all the webinar. So you don't have to wait until the end of the webinar. Just ask your questions, and then we will try to cover them at the end of the session. So I think with that, we can start with our first speaker, who is Dr. Sophie Schotter. So I think you can share your screen right now. And I'm going to mute myself.
Hang on. Sorry, bear with me a moment. Technical glitch. Here we go. Hopefully. Sorry, there we go. Perfect. There we go. Can you see my slides okay? Great. So, what are exosomes? And they really are, they're tiny, tiny, tiny little vesicles, these tiny messengers, but what they hold within them can truly deliver transformative results through a whole bundle of different possible contents. And this is what's important to realize, that not an exosome isn't just an exosome. An exosome, sorry, bear with me a moment. I can't talk at the moment. Sorry, my love. Sorry, I had a child come into the room. Sorry. An exosome isn't necessarily just an exosome. An exosome can come from many different sources, and it can contain many different things that in turn convey many different messages. So while everyone talks about the word exosome, that can actually mean many different things. So what we've seen over the last 10 to 15 years or so is this huge surge in interest in exosomes, a real explosive growth. But papers earlier than that are pretty sparse. We're now all talking about them in relation to not just regenerative aesthetics, but regenerative medicine as a whole, cancer medicine, drug delivery, and they're often studied for their biological signaling roles and also for their potential as biomarkers or therapeutic carriers. And if we compare that with botulinum toxin, which is maybe our most often performed treatment within a regular aesthetic clinic, we have this huge long-standing research history which dates back to the 1980s. It's well studied for both cosmetic and therapeutic domains, so dystonia, migraines, bladder dysfunction. And these papers are very focused on clinical efficacy, mechanism of the neuromuscular blockade, and side effect profiles. So we're still dealing here with exosomes with a very new area, not just of aesthetic medicine, but of aesthetic regenerative medicine, but of regenerative medicine as a whole. We're also seeing a huge surge in the number of trials being conducted. So there's somewhere around 300 to 500 trials ongoing at the moment, and most of them are quite early phase, so phase 1 or 2, so in the exploratory or first in human stages. So many of these are open label or non-randomized because of the fact that they're very early on in the clinical research process. And again, the most common indications for these trials are in oncology, orthopedics, things like cartilage regeneration, neurological recovery after stroke or spinal cord injury, skin and wound healing, and then also, especially in the early COVID era, there were studies around exosomes in COVID-19 therapy. Within aesthetics specifically, there are very few trials that exist. And again, they're mostly early phase. So focus areas have been melasma in combination with laser, skin rejuvenation, and the word I hate that unfortunately still is too often talked about, but anti-aging. And then some use of microneedling or topical application. And most of these have been conducted in Asia, in the Far East, or in the Middle East. They're pretty small scale trials, maybe 60 participants or less, and they're also quite short duration. So I hope this makes you appreciate that as much as we're talking about exosomes day in, day out at the moment, what we actually know about them really is the tip of the iceberg. There's still so much for us to learn and to understand. And if you're anything like me, that excites you, because the future of what we could do with these tiny little messenger bundles is incredible. So a little brief history. Exosomes are these tiny extracellular vesicles. So they're between about 30 and 150 nanometers in size, and they're secreted by cells. And originally they were thought to be cellular waste, but now we recognize them as these critical mediators in cell-to-cell communication, and that they have implications for so many different fields. So some key moments were 1946, and this is when researchers studying blood plasma noticed these tiny particles or fragments, but they didn't know what they were. So even though it wasn't known what they were, and therefore maybe wasn't about exosomes directly, it laid the groundwork by observing this unexplained cell-free particulate matter. And these early observations really helped to build the concept of this subcellular vesicle release. But technology at the time was nothing like technology today. We didn't have the advanced techniques like electron microscopy that would have helped to define or to isolate exosomes clearly. 1981 was actually the first explicit mention of exosomes, and this was used in literature to describe these vesicles released from the plasma membrane of cultured cells. So it really was this formal naming and description of what an exosome is as a distinct biological entity. And actually this was in relation particularly to cancer cells. And then in 2007 came the exciting discovery of microRNA in exosomes. And this was a publication of a real landmark paper showing that exosomes carry messenger RNA and microRNA and that these can then be transferred between cells. So this really revolutionized this field of exosome medicine. They were no longer just perceived to be these waste vehicles, but actually active vehicles that can communicate genetic information. So this opens all sorts of new doors for research into exosomes as biomarkers of cancer, neurodegenerative diseases, and also inspired the development of exosome-based drug delivery. So here we can see that they're a little bit about, you can see how small these tiny exosomes are in relation to other cellular structures. And here you can see them on the outside of the cell membrane, and then they're absorbed. They bind to the cell membrane wall, and they're absorbed into the cell themselves. But when they're actually produced, they're from these multivesicular bodies, which you can see these MVBs. So the MVBs are the kind of where the exosomes are born. So they're released by nearly every single type of cell in the human body. And their roles stem from or range from cell-to-cell communication, which I've touched on, but also immune modulation and potentially also disease progression. They're usually round or maybe slightly cup-shaped, and they're enclosed by a lipid bilayer, the same as a human cell. So these are rich in cholesterol, sphingomyelin, ceramides, phosphatidylserine. And this membrane provides protection and also selective targeting properties. So they can also display membrane proteins, which are important for cell-to-cell interaction. And inside, they can carry all sorts of different biologically active molecules, from proteins to the nucleic acids we've mentioned, lipids, metabolites, a vast array of different things. And that in its own right creates a recipe, an instruction manual from one cell to another. So the actual exosomes themselves can differ in size, composition, and in terms of function, but they're broadly classified into, this is extracellular vesicles rather than exosomes. So extracellular vesicles come in three different sort of classifications. So exosomes are the smallest of these. So as I said before, 30 to 150 nanometers in size. So they're released by these multivesicular bodies, which I showed, and they're the most studied of all of them. We then have microvesicles, which are bigger, 100 to 1,000 nanometers, and these bud directly from the plasma membrane. And then there's also another type of extracellular vesicle, the apoptotic body. And these are, again, bigger, 500 to 5,000 nanometers. And these are released by cells which are dying, so through apoptosis. And they can actually contain cellular organelles as well as nuclear material. So why all the interest? These are complex structures, far more complex than what's been used for drug delivery previously. And one of the most common questions I had in the early days of talking about exosomes was, what's the difference between an exosome and a liposome? These are really complex structures that have a far more involved role in the human body and in human biology than a liposome does. They really do facilitate this intercellular communication and the delivery of different substances between cells. They're part of so many, I mean, I'm not going to say quite all, but very, very many processes in the human body. We can also use them as biomarkers in various diseases, and then the drug delivery. And I think it's really interesting that within aesthetics, we talk about exosomes as a therapeutic tool, but actually they're also used quite widely, more widely, as a diagnostic tool. So both the therapeutic and the diagnostic applications rely on the exosome's ability to carry these biologically active molecules. And therein, by doing that, they're reflecting the state of the cell that they were born from. But exosomes in therapeutics are what we're using in aesthetics. And this is actually this tiny part of exosome medicine as a whole, because about 70 to 80% of the applications of exosome at the moment are actually in diagnostics. And this is because they carry this molecular cargo which reflects their cell of origin. And these are perfect biomarkers, especially when we're talking about things like non-invasive liquid biopsies. But yeah, there's so many different applications that we don't yet understand. And the fact that we have such a small fragment of the market, 20 to 30% in the therapeutic space, means that the potential for that to grow is huge. But at the moment, a lot of these applications are still experimental and not clinically, not approved for clinical use. So the key mechanisms and steps of action of exosomes, first of all, is their creation and their release. So they're formed within the cells, within these multivesicular bodies I showed earlier, and then these fuse with the plasma membranes and release the exosomes into the extracellular space. They then travel through body fluids to reach wherever they're meant to go, and their surface proteins and the lipids help to protect whatever's inside and help them remain stable. These surface molecules then mediate binding to target cells, and they can enter through different processes: endocytosis, direct fusion, phagocytosis, and then once inside, they release their cargo. So all these different things we've talked about, the proteins, the nucleic acids, lipids, and that's released into the cytoplasm. And this cargo can then go on to do many different things, like modulating gene expression, for example, through gene silencing, which microRNAs can do. It can alter protein function or signaling pathways, and it can also affect cell metabolism or immune response. And how the recipient cell responds to that will also vary depending on the cargo and the context. And it could create immune modulation, it could promote tissue repair or regeneration, or it could improve neural communication. After it's been internalized, the exosome-containing endosome can do, again, many things. It could fuse with lysozymes for degradation, which can limit the functional cargo delivery. And in that case, the cargo is then recycled and reused. Or it can release the cargo into the cytoplasm. And when it releases its active cargo inside the target cells, it can have so many different effects. So the messenger RNA can be translated into proteins. The microRNAs can bind to messenger RNAs and regulate gene expression. So this is by silencing or degrading them. Proteins could activate or inhibit signaling pathways, and lipids or metabolites can modulate the cell membrane or metabolism. RNAs are these crucial players in the modulation that exosomes exert. And these are protected within that lipid bilayer so that they can survive the environment in the extracellular matrix or the body fluids and reach that target cell intact. And it's important to remember that the RNAs kind of trump whatever else is in there because they're true information carriers. They directly alter the regulatory gene networks of the recipient cells. So proteins and lipids can trigger signaling, but the RNAs provide this more sustained and programmable effect. But only a portion of the RNA inside exosomes is of known function, and a significant fraction, we don't really know what it does yet. Many studies have identified specific microRNAs and messenger RNAs inside exosomes, which have well-defined and characterized roles in gene regulation or immune modulation or disease process. And high-throughput sequencing reveals quite a complex and diverse RNA population, and this includes many novel microRNAs or fragments, as well as long non-coding RNAs and circular RNAs, which we don't know the functions of yet. So many of these roles are still under investigation at the moment. We have to understand that there is still so much that we don't know. And the exosomal RNA profile is highly heterogeneous. It varies dependent on what the cell of origin is, and also our physiological state or our pathological state of disease. Many of these RNAs can be loaded passively, or they serve roles, maybe regulatory or structural roles that we don't understand yet. But the most important RNAs in exosome cargo are that messenger RNA and the microRNA. But we have to understand that those roles are quite distinct from one another. So as I said earlier, the messenger RNA, which is the one telling a cell to make a protein, and the microRNA, which is silencing a production of a specific protein. I think it's amazing to realize that this is such revolutionary science, that the importance of microRNAs was recognized in 2024 by, so the scientists who discovered it were awarded the Nobel Prize in Physiology or Medicine. So I think it's really incredible to realize this is absolutely at the cutting edge of science. So just again, as an overall of the things that exosomes can do, they really are these vital mediators, and they influence so many diverse processes within the cells. So we've touched on a few of them, but angiogenesis is another one very relevant to our practice of aesthetics, wound healing and immune balance. So for me, it's something that I'm already excited by, but I have to say I'm even more excited about what the future can bring with this. And I'm going to hand over now.
Okay, perfect. Many thanks for your introduction on exosomes. So I think, Hernan, if you can share your presentation, we're going to move to photobiomodulation, autologous exosomes and their context a little bit. You are muted, Hernan.
Yeah. Now you can hear me. Yeah. And I'm sharing the screen. Let me know if it works, Laura. You can see it? Okay. So basically, this is the disclosure Dr. Cordero already talked about. We're going to talk about some evidence and some of the things the MCT system can do. Of course, you can find a lot of evidence on the internet, on PubMed, about what light does, what temperature does in terms of growth factors, of more physiological content of PRP, of pH, etc., etc. You can really take a look. It's very well documented. Photobiomodulation is 80 years old or even more. There have been many attempts in the past. The difference mainly with people just illuminating syringes, and what MCT does is the control of virtually every variable that can be controllable in terms of energy output, absolutely anything that has to do, that means controlling light, anything that means controlling temperature, and of course some physiological things, some physical things, as turbulent flow of the sample, etc., etc. So many things to be controlled, but this is the difference between illuminating a syringe, as I told you before, and really having a treatment and changing energy and change the response of cells, etc., etc., etc. Some of the variables are very difficult to control. As a matter of fact, light, you need exquisite control. If you release a little bit more energy or a little less, you will be out of the sweet spots, and then you will have no action whatsoever. So this is basically what MCT does. It controls the energy that we can deliver, so that you can really get the amount of energy you want everywhere and absolutely in every corner of the sample. That's pretty it. It's photothermal biomodulation, but it's a very clever way to do it so that you can perform it in your consultation. So basically, our system can stimulate any product that is in a liquid phase and it has anything alive inside to be stimulated. Of course, we are focused on PRP and any derivative from plasma, but people are doing all sorts of off-label applications with fat and with all kinds of autologous liquids. The general idea is to obtain the autologous material, to stimulate it by the MCT system, and to re-inject it in the product, in the patient. We are 100% autologous products, and no additives, no chemical, no whatsoever. This is exactly what the patients are looking for right now. So the system can control temperature from 4 to 45 degrees and light, different wavelengths. But the key of this system is that we understand how to work with temperature and light. Basically, they are completely different in their conception. Temperature stimulates all sorts of tissues in the same way, but the reaction it produces is completely different. This is opposite to light. Light will only stimulate certain structures, certain tissues, but it will get always kind of the same action. So basically, let's take a look at some examples of, for example, what temperature does to platelets. It will change the shape from an octopus-like or a chaotic shape to a spherical shape. It will change the internal structure organization of the organelles. It will be like peripheral tubular layer of tubulin. But you will also see this on a chemical level with BASP complex going down. This is an inhibition marker, and the activation markers going up, glycoprotein to P3A, phosphorylation of P38. Basically, what this is telling us is that platelet gets primed for action. Platelets are ready to work, and you can see this at the end of the journey with clinically, because platelets really work much more and better with more ATP, with more growth factors, releasing more exosomes, etc., etc., etc. Another action of temperature, for example, with a different thermal modulation protocol, is in exosome release. Temperature will act in 3 different levels, enhancing membrane fluidity, enhancing kinetics inside the cell, so everything is colliding more. More collisions means more docking means more release of exosomes. Every single of these processes play a role to obtain a higher amount of exosomes. And of course, a third, let's say, a third process or a third step in this crucial exosome release is the reduction of tethered exosomes. Tethered exosomes are the exosomes that get stuck to the outer side of the membrane once released. So, if we add all these actions all together, you will get the huge amount of exosomes we are able to obtain with MCT system. We are going to talk about this specific number later. What happens with light? Photobiomodulation. Light will only interact with chromophores. Chromophores are structures that change chemical energy to electromagnetic energy to chemical energy. This is what happens in our eyes when we see or photosynthesis with plants. Basically, we have chromophores in all our cells, and they will produce second messengers, and through transcription factors impact on different processes. Like here you can see wound healing, angiogenesis, antioxidation, etc. But specifically, we are working on a couple of chromophores. Number 1 is opsin. Number 3, there are 1, 2, 3, 4, 5. Number 2 is the rod opsin that you can see it in our eyes. You can have it in our eyes. But this is number 3, is sensitive to blue light. It's increased through transport through channels related to voltage. They will increase the amount of calcium, hypersensibilizing cell. Another chromophore that we are always working with are the macrocomplexes of phosphorylative oxidation. Macrocomplex number 1 and number 2, these are the cryptochromophores. They are sensitive to blue light. And number 4 is sensitive to red and infrared light. This is cytochrome C oxidase. You know this. This is a translocation of protons. Protons get back through a pore in ATP synthetase, And then while it rotates, it will produce a huge amount of ATP. ATP for cells are like money in our pockets. It will play a role in activation, in action, in whatever work the cell must perform. So basically, MCT is a system that will deliver tailor-made secretomes. This is basically it. These are all secretomes with different characteristics. You will have a higher amount of growth factor, anti-inflammatory proteins, a huge amount of ATP, or what Dr. Schotter told you about secretomes. And this is exosomes. And this is the point we will be addressing right now. Exosomes, as you were said before, these are communication between cells, but this is not a phone call. This is more resembles an explosion. You know, I send all my exosomes away, and everybody will get them. Then the utilization of what's inside exosomes will be determined by every cell. If they want to use it, this will undergo the back fusion, which is the slide Dr. Schotter showed you. If they are going to be wasted or recycled, they will be fused with lysosomes. This is very simple, but you need to understand that we are talking about autologous exosomes. This is what we do with MCT. We are not a cosmetic exosome. Right now, there are more than 400 companies selling products, cosmetic, and all of them are saying that they are better than the other one, just to the side of them. These are non-autologous, these are non-injectable, these are cosmetic. We are a medical device, class 2 medical device, worldwide certified. This is a completely different game. This is a big umbrella. It's called exosomes, but this is a completely different product. This is a medical device, and of course, it's a medical treatment. So if you want to analyze, if you're going to study about exosomes, you will find 5 different let's say columns. Number 1 are exosomes products that are not legal. We're not even going to talk about this. But then you can make the difference between non-autologous and autologous. In non-autologous, there are the ones that are people are trying to inject them, where, you know, blinking an eye and saying nothing's going to happen. Well, forget about these. These are completely not safe because they haven't done the studies or the certifications. They want them to you to do it for them. So forget them. If they come from plants, from animals, from other humans, they can only be cosmetic and they can only be used topically. So number 1, cosmetic exosomes. Number 2, non-cosmetic exosomes. These are true autologous exosomes, which is like MCT. And of course, we are a medical device, we are safe, we are worldwide certified, etc., etc. But there are also a huge amount of product right now. They are autologous, they are certified, but they are not effective because they are these PRP companies or you know, people that have been selling autologous material for a long time, and they say, oh, now we have exosomes, and they give it, they have a blue triangle, they paint it red, and they say now it's exo triangle. Now we have 55 million here, but in that country we have 70, and we have 54, whatever. So this is really complicated. So I just published. It's not published yet. It's waiting for acceptance. A classification, to try to put all this together and to explain to people what's the difference between an autologous, worldwide certified medical device and a cosmetic exosome that you get it from a goat, and you are trying me to inject it. So this is a completely different scenario and a different game. You will get all sorts of claim. If companies that are selling human exosomes, they will tell you that autologous exosomes, they are from old people. This is not true. Older patients will release more exosomes than younger patients, etc., etc., etc. All these are claims that they are not true, and they are trying to. There's a lot of people that are right now working for confusion, not for transparency. So when you try to approach this exosome world, you will find three main areas where people are talking nonsense: misleading marketing claims, problems with biological safety and efficacy, and regulatory framework. So we are going to talk just a little bit about 4 or 5 of the main things that are crazy right now in the world of exosomes. Number 1 is the source. People are saying they're getting exosomes from a cow, from a horse, from a plant, from a bacteria, from another human being, etc. As Dr. Schotter said before, exosomes are nanoparticles that have a membrane and have a content. The membrane is a bilipid membrane, but it has a lot of proteins inside. It has tetraspanines, and of course it has major histocompatibility proteins. So, exosomes are antigenic. They are, of course, not as antigenic as cells, but they are antigenic, and they will cause problems on the long run. Many things that we don't know so far. So when somebody says, hey, I have a cousin that is injecting them in Japan and blinking an eye and saying nothing's happened, don't worry about it, forget it. You are physicians, nobody's going to protect your patients but you. So if it's not certified, if it's not autologous, don't inject it, forget it. Number two is the load. This is what Dr. Schotter told you about. This is the same slide. 1/3 of the content is well known, 1/3 of the content is predicted, and another third is unknown. If you work with autologous exosomes that are naive, like the ones we use in MCT, they are already there, they are circulating. We have 30 years of millions of treatments of PRP. These are the same exosomes, they are there. And nothing ever happened, zero incidence of cancer, zero incidence of whatsoever, no side effects. Then you're completely safe. But if you want to do the same with other people exosomes, with goat exosomes, with a sequoia exosome, that's up to you. Nobody knows what's going to happen. Another thing that's very misleading is people are telling you this is a tissue specificity from the exosomes. I can send it to your hair so it grows. If you are a tennis player with your left hand, I can send it to your left elbow. This is not going to happen. This does not exist. As a matter of fact, tissue specificity is a stupid thing because what we need is a cell specificity. I don't want the exosomes on the dermis. I want it on the fibroblasts. There are 7 or 8 mechanisms to uptake exosomes from the cell. 90% of them are non-specific. So every cell are going to get the exosomes. I cannot send it to wherever I want. The only way of sending it there is by using a needle. And you can use a needle only if you are a medical device, injectable, certified medical device as MCT. Another thing is the number. People are talking and they're crazy about the number. I have 30, I have 40 billion. They say whatever. They don't back it up. We have published it. We have retained this number for 1.5 years. I didn't want to publish it because this is not the most important thing. We are around 300 billions will be per ml. 299.6 billions per ml. And with MCT, we use 10 ml. So imagine the amount of exosomes we are using. Some other people are doing, propaganda or advertising with 30 billion. We are talking about 1/10 of our dose. You have to scratch the surface. You have to read the tags of the products, and you will see how crazy is this right now. The most important thing is the quality. This is why I don't like to talk about the amount. The quality. We have taken videos, pictures, measured the site, evaluate cluster, differentiation cluster of surface, and more than we are doing right now. We are releasing a new study with Dr. Cordero very, very soon. Well, people haven't done none of this. If you take a look at the exosomes labels of the product, many of them are not even exosomes. They are products of isoleucine or biotin or whatever. And so they have one excipient that has an exosome, and so they will call it exosome product. It's nonsense. It's an insult to physicians. So please read all labels, ask all the tough questions to everybody. The big picture of the legal scenario, at least in Europe, is very simple. Either you can use a cosmetic product as topically, or you can use autologous product that you can inject. And then here you have the ones I told you before, only marketing product and as a science-based medical device. This is very simple to understand. Many people don't like it, but it's very simple to understand. We are doing all these kind of webinars and a lot of academic actions. We are publishing books. We are members of international societies for extracellular vesicles. So we like to show you a very big difference between what we do and what is now being done by many other companies. So we have a lot of technical documents. These are not commercial. These are all technical documents. And you will see some of the studies. If you check on the studies of different companies that they say that they are top of the, some of them don't even have a single paper. They have a letter to the editor or whatever. This was the analysis I had to do in IMCAS this year. I was invited to talk about this, how poor is the study production of these companies. You can check You don't read all of this, but you can check here the number. These are 10 papers already published, 20 papers, 17 published, 3 already submitted, some others on fieldwork, etc., etc. We are now engaged in 37 studies worldwide, and we'll keep it going. So, this is a physician-to-physician company, a scientist-to-scientist company. Right now, we are empowering all type of study. Many of them are clinical trials in different places of the world and tackling different specialties. If you want to take a fast look at this, I think we can talk a little bit about Dr. Hernandez, 7 cases with systemic autoimmune disease with neck rejuvenation, the same as Dr. Beltran, 45 patients, neck rejuvenation. Professor Mercuri in Milan, linear morphea, more sessions that normally they use 2-3 sessions, and this doctor used 5 sessions, but spectacular results with a very difficult pathology. Dr. Tejero, Professor Tejero with hand rejuvenation, Professor Naranjo with hair shaft survival for hair transplant, you name it. Professor Melamed, growth factor content, Professor Kosarev with Bell's palsy, paralysis, etc., etc., etc. Dr. Amesti, wound healing. This is a 96 different hour, one single session of exosomes. This is an 85-year-old diabetic patient. This is a very interesting case. My reviews, you can download them at PubMed. Professor Mesana using FAT, Professor Delia Vila, Dr. Avila, Dr. Cordero, different basic studies, clinical studies. And this is the one I was telling you about, Dr. Cordero study with 299.6 billions per ml. This was the measurement. And you can check on some cases, some clinical cases, of course, acne, they are very eclectic group of pictures. Face rejuvenation, periocular rejuvenation, hand rejuvenation. This is all aesthetics. But we have also these eumelanic properties of exosomes that sometimes are not very well understood. When you have real melanosis, they take it down, and when you have a hypopigmentation from a neuralgia like here, they will take it up. There is not enough evidence so that we can protocolize this, but we are having a huge amount of case reports, and we will definitely have a protocol handling melanin in the future. Some cases, spectacular cases about psoriasis and, of course, rosacea. These are great cases. You will also find wound healing in all sorts of specialties. The one to the left also had surgery, but we are really very happy about this. This is a very important pathology to the right with nothing but exosomes, and to the left, superficial lesion. Some problems about when injecting polylactic or when injecting hyaluronic and having vascular compromise. So, everything that in gynecology, we are having great success in 2 or 3 pathologies: vaginal rejuvenation, lichen sclerosus, remarkable results. And ovary activation, we are conducting a clinical trial so far, 10 cases, 100% pregnancy. We are very happy about that. And of course, androgenic alopecia. This is very well known. We have remarkable results from left to right: Valencia, Iraq, Rome, Serbia, Russia. So it's very very simple. Some figures, and I think we will call it a session. We are present in almost 70 countries, more than 120,000 treatments. 120,000 treatments. Yeah, I did it correctly. I said correctly. More than 1,100 machines worldwide. And the most important, at least for me, slide is this one. This one is the one that are telling you how safe and how effective this product is. And this is something that you can translate directly to your patients. Worldwide certifications, very strict certifications that are saying that we are safe, we are class 2 medical device everywhere. It's not that we are just MDR, class 2 medical device, and then somewhere else we are cosmetic. No, this is injectable medical device worldwide. So thank you very much. It's a pleasure being here with you today. I will stop sharing my screen, and I guess we can have a great Q&A session. Dr. Cordero, I leave it to you.
So I'm going to talk a little bit more about how to, in practice, bear with me, let me go to the right slide, how we use the MCT in clinic. Here we go. Perfect. Oh, sorry, it's restarted from the beginning. Here we go. So the first thing I would say is that the exosome space, because of all this excitement, is becoming increasingly crowded, and you will find patients, consumers who know the word exosome but don't necessarily understand the differences. And as Dr. Pinto just explained, we have something here that is so safe, and I think this is a real opportunity, because certainly in my practice I see more and more people who are worried about the fact that the aesthetic space is becoming so crowded with products of very variable quality, without the data to back them up, and a lot of marketing. And so they're worried about having things injected that they don't really understand. And for so long we've had PRP, which, I mean, it can be great, but there's a ceiling on what PRP can achieve. And here we have something that creates a new level, but is 100% created from that individual's own body. And I think that's a real opportunity at the moment. So a very simple 3-step process to how we create the MCT plasma and or exosomes in clinic. So step 1 is that you will take your blood sample from the patient, or it can be a fat sample as well, although that's not an area within which I practice. So for me, it's a blood sample, and I create PRP. So I use the system that I've used for many years, which for me is a very good, very safe system that we know has a good platelet yield and minimal contaminants. So that's what I do, and I create that as per the protocol that I would use to create normal PRP. I then insert that PRP that's been created into the MCT kit. So generally I would recommend getting a 20 ml PRP sample if you can. That would be ideal. So we're going to insert it into the PRP kit, and this is a really simple process. So the kit itself looks like, you can see the little tray that you can see here, and there are 2 clips on there, and you can see on the left what it looks like when it's open versus what it looks like when it's closed. So what you're going to do is when you're inserting it, you're going to have the caps open, you're going to have the caps facing upwards, and you're going to inject with either a non-Lurlock or a Lurlock syringe into it. And then when you extract it, you have it the other way up. And oh, hang on, where was that? No, I didn't miss it. Here, sorry, I think I did miss a video. Here, let me play these videos for you. So here, and then here we have the MCT units itself, a very, very neat, and I mean, actually, I think this picture makes it look bigger than it is. It's actually a very compact unit that stands on the side in my clinic. It doesn't occupy space. And we load the tray into the unit itself, as you can see here. So you then select the protocol you want to use, whether you want to create MCT plasma, or whether you want to create MCT cells or the exosomes. And it's all preset in there. So you have everything set up, you put it ready to go, and then afterwards you can extract it and administer it. And as we were talking about, you can inject these. because it's not a cosmetic product. It's not something that has to only be applied topically. You can inject this. So an extremely simple kit. And I would say that most commonly, for me in my practice, I would combine using both MCT plasma and MCT exosomes because it's actually very easy to do from a single sample. You would create your MCT plasma first, and then while you're administering it, you do a second protocol to create the exosomes, and then you can administer those afterwards. So you have a very, very simple way that you can actually give patients the best of both worlds. And each administration takes maybe 10 to 15 minutes. Now, of course, you need to allow a little bit longer in terms of real time in clinic because you want to take the blood sample and then you have to create your PRP from that through centrifugation. So the whole thing gives you a real opportunity to spend time and interact with your patients as well. So for me, it's a real, as I said, a real opportunity that we have here to be at the forefront of a new wave of regeneration. I think regeneration is where it's at. And that's not because I am not a fan of botulinum toxin or fillers. I still love them. But I think what we're understanding now is so much more about how and why we age, how much, what goes on physiologically in disease states. And we know that we have an opportunity to modulate that in a way that we never have had before. And for me, everything that we do as aesthetic physicians should start with health. And the health of our body's largest organ, the skin. And here we have something with which we can really change how that skin is behaving. We can combine it with so many different technologies, whether that's microneedling, whether that's a light laser treatment. You can use it in the way you would other exosome products, but you have something here that's exceptionally powerful, that's autologous, but you can use it to enhance healing, to reduce downtime and inflammation. And I really think this is something our patients are crying out for. I can attest to the first Instagram post, which I made about MCT, the amount of messages that I got afterwards from both patients and practitioners actually interested in this technology was astounding. And it's really led to a huge wave of inquiries in clinic about it. So as I said, you can use it for post-treatment recovery, any sensitized or inflamed skins, and that doesn't just have to be post-procedure, that could be someone with a compromised skin barrier. Early aging or prejuvenation, and again, also for treating acne scars. Those are kind of the key indications where I would recommend using it. Now, one thing we always talk about with PRP is platelet quality. And, you know, I am as healthy as I am. And so my platelets have a certain quality that's reflective of my health. And what we can say is that the MCT machine will take what you have and amplify its potency, its power. But of course, the healthier your individual, the higher quality their platelets, the better their platelet function, the better the results you'll get. But platelets are also produced relatively rapidly. Their lifespan is under a month. So you also have an opportunity to talk to your patients about lifestyle and optimize their health. And then potentially, although it's something we can't measure outside of some hospital settings, improve their platelet function further. Age, obviously, can impact. Certain comorbidities, medications can impact on our platelet function. Lifestyle, as I just hinted at, which we can modify. And then obviously the condition we're treating and how severe it is. And expectation management amongst our patients is always so important. I always under-promise and over-deliver. That's my motto. We've talked about safety. I think, again, huge patient concern at the moment. We're seeing, at least in the UK, a tide of problems from aesthetic treatments, and people want to know that they're in safe hands. When will they see results? Typically around 4 to 6 weeks after treatment, although they may get some anti-inflammatory benefits earlier. And I would not talk about this as a volumizer. It's not it's not an immediate corrector. This is an all-over regenerative tool, something that is going to improve the health of the skin as a whole, rather than treating focal defects. So for me, it's been a really easy integration into clinic. I find nowadays the majority of my patients are focused on regeneration over wanting to fix the surface signs. And that's certainly how I've practiced actually for many years now, talking to my patients about deeper aging. And so MCT for me plays so perfectly into that. I take my time explaining how it works and why it's different and what the benefits of that are. You can, in a single treatment session, inject 2 to 4 mils of autologous exosomes same day. And as I said, I often combine it for post-recovery. 3 sessions in total, spaced 4 weeks apart, and quarterly maintenance as needed. I find documentation, you can use Visier. I personally use a device called Aura, but using some sort of skin quality device that I think is particularly helpful, because we all know it can be really hard to photograph skin quality in a 2D photo. And my advice to you, if you're looking at starting out, is to start small. Don't try and treat everything. Get comfortable using the MCT for one particular indication, or maybe a couple that you're going to do a lot of. And then as you're comfortable with it and you see how well it works and you trust the results you're getting, then expand into other areas. Make sure your staff understand it. And actually, I think it's great for you to treat staff as well. Get them to experience the treatment and the benefits. Managing your patients' expectations is the most crucial step to happy patients. There's no such thing as a miracle. And what we have to understand is our patients need to understand the difference between the health benefits that they're going to receive and what they're going to see in the mirror, and that they're not going to suddenly look like they've had a surgical facelift, but the results will be there and they'll be tangible. And then making sure you have regular touch points to track the results that you get. So this isn't an instead of anything that you're already doing in clinic. It's a real complement to and an addition to your portfolio. Thank you so much.
Thank you to both speakers. I'm gonna put this view right now. Okay, and I think we got many questions, but first, that's good. First, I wanted to start a little bit with the title that we gave to this webinar. So what do you think? Are autologous exosomes truly a game changer in aesthetic medicine? Or do you see them more as a current trend that will somehow plateau in the end? So what do you think from both point of views?
Do you want to start, Sophie, or shall I start?
I think it's a step change. I think this is something really exciting. And I think we will only see more innovation and more understanding and more applications as we go into the future.
Yeah, I think the same. This is a game changer, but right now, autologous exosomes are leading the way and they're delivering safe and true results. And right now we have a crazy trend with a lot of products that are not fulfilling certifications. And so a little bit of both. It's a trend. There are a lot of people trying to surf the wave, but some products will remain, and definitely exosomes will give us a huge amount of satisfaction in terms of regeneration and health in the future.
Okay, perfect. Also, how do you think autologous exosomes compare to other autologous aesthetic treatments such as PRP, fat grafting?
I think for me, this is different. This is the potential to amplify PRP and take it to a different level. You know, a 3 times multiplication in growth factors. The fact that we can create exosomes from a PRP sample or amplify exosomes in a PRP sample. So for me, it's taking PRP to a different level. And again, fat grafting isn't something I practice. I think it's amazing, but again, the MCT machine has the potential to amplify an adipose sample as well. I think this isn't replacing anything we have available. This is taking it to new levels.
Yeah, absolutely. And right now, people are, as I said before, facing off-label indications of whatever. They're it's so safe, it's so effective. They are trying it on and everything. But I think the real breakthrough, it's not in the short term, but in the midterm and above. In the short term, you will have a much better PRP, more hydration, more turgidity, or more basically more aesthetic satisfaction. But this is not the main benefit of the treatment. The patient will come back at 1 month control and say, yeah, it's great, it's a better product. And they will come back at the 2nd month and they say, I cannot believe it, because it is cumulative, and then it really regeneration takes place. So in the mid range and above is where it's really a remarkable treatment. It's really a breakthrough.
Okay, thank you so much. I'm going to start with more specific questions that we received from the audience. Okay, we are being asked if are there any cases of use of MCT exosomes that has allowed patients to discontinue any combination therapies such as minoxidil, finasteride, light therapies. I don't know, maybe Hernan.
From my side, no problems whatsoever. No contraindications in terms of sensibilization or allergies or disruption of former medicine or treatments. No, it is cumulative. You can add it on top of whatever you're doing. Of course, this has to follow a medical criteria. The treating physician has to decide that this is a good option. But yes, we are having awesome results and zero problems into this regard.
Okay. We've got another question. So do you always inject these MCT exosomes?
You can apply them topically, you can microneedle them in, but personally, I, out of preference, will inject them because I have something here which is safe to inject and I want the maximum exposure of the tissues to those exosomes. So if I can inject it, why wouldn't I?
Yeah, I agree 100% that there is a study from Dr. Amesti. It's published. She used it topically because now there are people approaching this product with occlusive bondage on top, etc., etc. But in any case, my opinion is the same as you, Dr. Ashotar, as you, Sophie. I will always inject it because this is a medical device. It can be injected. It's the real difference.
Okay, so we know that they can be injected, so we can use it that way. And we've got many questions about comparing exosomes, so autologous exosomes, with other treatments that already exist in aesthetic medicine. For example, somebody is asking us, how do we compare exosomes with other phototherapies like low-level light therapy or even with polynucleotides? So what are your thoughts on that?
They're different. You kind of can't compare them. You're comparing, to take a polynucleotide analogy, a salmon and a trout. They're not the same. They may all fall within a spectrum of regenerative therapies, and you can use them in combination, but they're not an instead of.
Yes, some people love the fact that they are natural. There are no additives, no chemical. This is what right now people are doing. We tend to do this. We compare it to everything. Now I want to compare it to a brick, and I need you to explain to me why this is better than a brick. No, it doesn't work like this. This is an awesome treatment that you need to add it to your armamentarium. It has a place. MCT has a place on every consultation. And, according to what you see on your patient, what your patient wants and what you think it's your criteria, you will be able to use it virtually on any other treatment where you use a needle, because this empowers regeneration and reduces the downtime and the damage. So I don't think you should compare it to all these things. You can, but try it. You will see the difference.
Okay, thank you so much. And we've got also several questions on whether can MCT system be used with platelet-rich fibrin, and are there any specific program to prepare the PRP that we use with MCT system? So basically the amount of vials, with which anticoagulant we are using, centrifuge and everything.
I'll let you take that because you've got more oversight of the different systems. Yeah, sure. At the beginning, we were thinking not of using PRF because we thought it might clot inside the cassette, etc. But it's not happening. People are. We are not recommending, at least at the beginning. But people are, as I told you, they are trying everything. The results are so good that they want to use MCT with whatever they are using in their practice. So if they are using PRF, they want to add MCT to that. They have tried them, and so far, no problems. So I think, yeah, it's fair to say you can use it. Second, in terms of what PRP, well, this is stimulating your platelets. So what I want is give me the best PRP you're able to get. We can discuss this. Some people like one brand, another people say, no, I don't like PRP. I don't get into this. Give me the best PRP you're able to get. If we have a good product, you will have an awesome product coming out. If you put garbage in, garbage out. So, this is important. It doesn't mean you need to centrifuge more or centrifuge. No, do it exactly the same way you are doing it. And in terms of anticoagulant, now there is also, people like to discuss about everything, but we have been using anticoagulant for 30 years. Sodium citrate, 3.8%. This is standard anticoagulation procedure, and it works remarkably good. There are some brands that are not using anticoagulant. They have a specific protocol, slow centrifugation, etc., etc. I don't get into this. If you are able to produce top quality PRP, I don't care how you get it, and you can feed MCT with this, and you will have a super product to treat your patients.
Perfect. So we received also, this is very, the webinar is very focused on aesthetic medicine, but we got many questions on orthopedics. So maybe, Hernan, you can answer. It's just that, has it been used in orthopedics, and in which indications, and whether we have any studies that show that we can improve cartilage outcome? Yes.
Thank you for the question. I am not a traumatologist. This is not my field of expertise. I can tell you there are a lot of people using it, mainly on osteoarthritis. I don't know any other pathology they're using, but I know that some clinical studies are being conducted as we speak. So I think we're going to have a lot of indications and a lot of information in the months to come. I really cannot answer too much. We have a lot of positive evidence, but these are not protocolized. These are not studies. People are saying, I just use it like this, use it like that. I'm having great results. Of course, the protocol is the same one you're using with PRP in terms of only 2 mLs or 1.5, etc., etc. But people are very happy with it. I don't know if the clinical trial we are sponsoring or helping with is tackling the cartilage regeneration. I don't know exactly what variables they are leveraging, but in any case, we can keep this contact and let you know, because for sure we will have a lot of news in the months to come for traumatology and for gynecology, for sure.
Okay, thank you so much for sharing a little bit of MCT in orthopedics. And so regarding also into this field or topic, so what do you think, just your opinion, that we will see the biggest impact of exosomes in the next 5 years?
Ooh.
Well, I think, sorry, Sophie, if you want, I can take this at the beginning. I think that traumatology is really going to explode, because what we are seeing is incredible. But we are running into results that will become indications in the future that were completely unknown or unimaginable. For example, as I told you, ovary reactivation, and this kind of indications. Now they're working a lot with erectile dysfunction in urology, and more things to come, autoimmune diseases, etc., etc. So I really cannot tell you what is going to be the most incredible indication of exosomes, but I'm sure we are going to hear a lot, but I mean a lot in orthopedics for sure, and wound healing. If you think about how good are the results of MCT in wound healing, and wound healing is transversal. Virtually every treatment we perform creates any sort of damage. I really it's difficult to foresee what's going to come.
Yeah, for me, where my practice is so centric on I'd say aging. I think I think for me the longevity benefits in the and I look at it as skin as our largest organ. Its health impacts our systemic health, and so I think that's super exciting. But I think the applications will see far beyond that, but I can't say which area I think will grow the quickest because I don't have enough oversight over exosomes as a whole.
Perfect. So, okay, we got like more than 70 questions. So, of course, we are not going to cover all of them. I just got crazy reading everything. But I think it's really good if you can summarize in one sentence or two sentences, like why should practitioners consider adding autologous exosomes into their toolkit?
Remarkable regenerative power. Autologous, 100% safe, worldwide certified as a medical device, and huge innovation. This is at least from the side of MCT. I don't know, Sophie, what you think.
I would say absolutely all of that, but it's also something that patients are looking for right now. So that's the only point I would add on to the end.
Great.
Thank you. So thank you so much. I think it's a bit late. We had a great discussion. So also presentations. Thank you so much, Dr. Schotter, for joining us. Your presentation was excellent. Thank you also, Dr. Hernan Pinto, for your presentation. You went really specific on MCT, and I think that's going to be very useful for all the assistants. And just thank you so much, everyone, for joining us today. We really appreciate that you took your time, join us, ask all of these questions. We know that there is a huge amount of excitement with MCT. So thank you so much for joining us. And I think we can hope to see all of you in our next webinar. Thank you so much,
everybody.
And bye-bye.
Cheers. Bye-bye.
Bye.